The overall goal of this screening procedure is to identify novel bioactive nanoparticles that can inhibit toll-like receptor signaling.
This method can help efficiently identify novel nanoinhibitors targeting toll-like receptor signaling in innate immune cells. The main advantage of this technique is that it is fast, robust, and easy to perform using reporter cell systems.
The implications of this technique extend toward the therapy of many inflammatory diseases because toll-like receptor signaling is found to be involved in the pathogenesis of these diseases. Though this method can provide high-throughput screening on peptide gold nanoparticles, it can also be applied to other nanoparticle systems as well as therapeutics, small molecules, and biologics.
To begin this procedure, prepare the culture mediums as outlined in the text protocol. Next, add 100 milliliters of ultra-pure endotoxin-free water to a clean 125 milliliter glass flask. Add 1 pouch of secreted embryonic alkaline phosphatase substrate powder and swirl the solution gently until it is fully dissolved. Incubate at 37 degrees Celsius for 1 hour to ensure complete dissolution of the substrates. Using a 0.2 micrometer membrane, filter the solution. Store at 4 degrees Celsius until ready to use. Next, add 1 pouch of luciferase substrate powder to a 50 milliliter centrifuge tube containing 25 milliliters of ultrapure endotoxin-free water. After the powder has dissolved completely, store at 4 degrees Celsius for up to a week until ready to use. Then,